How you doing, guys? Welcome to another episode, exclusive, higher level biochemistry, volume 7, proteins and enzymes. Let's go! Alright, proteins and enzymes. We need to talk about enzyme inhibitors, amino acids as buffers, and then we look at some UV spectroscopy analysis. IB understandings and applications. We need to discuss the role of inhibitors. We need to talk about amino acids. We look at some protein assays. We need to look at the graphs of enzyme inhibition, which is a common question. We talk about the enzymes, and then we look at how you could work out the amount of proteins in a sample. So in the standard level material, we looked at a lock and key method for enzymes. But there's also another method called the best fit or the induced fit model. That's where the enzyme essentially changes shape to make the substrate fit perfectly into its hollow. So that means that the enzyme can be continually reshaped, and it's able to shape in and around that substrate and have the interactions to produce the products, as that little diagram shows on the left. Then the enzyme will return to its original form and then be able to catalyze another reaction. Now, an inhibitor is an important molecule that plays a role in regulating the activity of an enzyme. An inhibitor is a substance that prevents the enzyme from doing its job, and the inhibitors work in one of two ways. So here we have an enzyme and a substrate, and the substrate fits perfectly inside the enzyme. Now, if we have a competitive inhibitor, that's one that comes in and fills the active site of the enzyme. So the substrate can no longer get access to that enzyme because the inhibitor is already in there. It's competing for the active site. Competitive inhibitor. A non-competitive inhibitor has a slightly different function. It interacts with the enzyme at what we call an allosteric site. Now, that interaction with the allosteric site and the enzyme actually changes the shape of the active site. So when the substrate comes in, the active site has changed shape, so it will no longer fit into the active site of the enzyme. So that lowers the function of that enzyme. So competitive inhibition, that's where our competitor resembles the normal substrate molecule, and it competes for the enzyme's active site. That reduces the ability of the enzyme to catalyze the reaction, as there's physically something blocking the site. We can overcome that by increasing the concentration of the substrate, and the enzyme will still reach its maximum efficiency just at a decreased rate because it has to release the competitor and then have a collision with the substrate. A non-competitive inhibitor that impedes the reactions by binding to the allosteric site, which changes the shape of the active site. A non-competitive inhibitor will reduce the effectiveness of an enzyme and lower its maximum rate of reaction. So we have to look at graphical analysis of enzyme activity. And what we have is a simple graph of rate of reaction and concentration. The enzyme shows a very particular shape. It's kind of linear or approximately linear for the first part of the process, and then as the concentration of substrate gets too great, the enzyme starts to peter off and it's not functioning at a linear rate anymore. Once the substrate becomes completely saturated, or the enzyme becomes saturated with substrate, then it can no longer catalyze the reaction any faster, and we call that the Vmax. Now, the Michaelis Constant Km is equal to the substrate concentration at which the rate of conversion to substrate to product is half the maximum rate possible. And this Michaelis constant gives an indication of the affinity of the enzyme to a substrate. So if you have a very small Michaelis constant, that means your enzyme has a high affinity for the substrate. And basically any substrate that's left over in the solution, the enzyme's going to grab hold of it very quickly. A large Michaelis constant means we have a low affinity for our substrate. That can tell you information about the rate of reaction. The higher the affinity, the greater the rate of reaction. The lower the affinity, the slower the rate of reaction. So the Michaelis constant can be found by looking at the Vmax and then dividing it by 2 and then going across to the curve, and that will tell you your Michaelis constant. The introduction of inhibitors into an enzyme affects the rate of reaction and it affects the graphs. We need to be able to determine the Michaelis constant of an inhibitor from the graph. So in red, here is our uninhibited enzyme reaction, and Km and Vmax have been shown for you. If we have a competitive inhibitor, it's in the blue, and a non-competitive inhibitor in the green. So we have to be able to describe the effect of the inhibitors on the enzyme activity. So a competitive inhibitor, it doesn't change the maximum efficiency of the enzyme. It will remain the same. The Michaelis constant, however, will become higher, which means it will have less affinity for the substrate. That's because some of those active sites have been taken by the competitive inhibitor and they physically can't get in there. A non-competitive inhibitor will lower the Vmax, so it will have a lower maximum efficiency, or it's less active. The Michaelis constant, though, for a non-competitive inhibitor will remain the same. It's not changing its attraction of the enzyme to the substrate, it's simply just lowering its maximum efficiency. So it's approximately the same. So it has the same affinity for the substrate, it's just that a number of the enzyme molecules have changed their shape. So this inhibition is an important way of regulating metabolic activity of healthy cells. One example of this is a product that is known as an inhibitor, where it is produced in a series of reactions. It's called product inhibition. So this is where we have a number of processes in a biological chain which forms a product at the end of the chain. So our substrate here goes through a number of different enzymes producing an end product with which this is this square shape. Now this square shape might be a non-competitive inhibitor for the original enzyme reaction. That means that when it bonds to that allosteric site, it's going to change the shape of the active site in enzyme 1, which is essentially inhibiting that reaction or slowing that reaction down. So this is called a feedback loop, because once that product is formed, it's going to start to slow the remaining enzymes down in the process. And that way we can regulate how much of that product is formed. We've talked a little bit about enzymes acting as buffers in previous videos, but it's important to remember that amino acids or proteins have a charge, and that charge can be expressed as a Zwitter ion. So when we add either an acid or a base to a Zwitter ion, the amino acid is going to act like a buffer and try and reduce that change in pH. Amino acids and proteins, they're amphoteric, so that means they can act as either acids or bases. So if I have my serine Zwitter ion, which would have an NH3 plus and a COO minus at the end, well, it can act as either an acid or a base depending upon what we do to this buffer solution. So if we added in some basic solution, some hydroxide, what will happen is the NH3 group, that is a weakly acidic group. So it will react with that alkali solution and try and neutralize it. So it would end up forming the amino acid with a negative charge. If we added that amino acid to a solution containing some acid, some H+, then the part of this amino acid that can react with the acidic part is the COO minus ion, the acetate ion. That is a moderately strong base, so it can accept a proton from the solution and form the carboxy group. The carboxy group is a weak acid, so it's essentially resisting some of that pH change. The final thing is to look at protein assays, which is determining how much protein is in a sample. And we can do this via using a technique called UV visible spectroscopy. Now, UV visible is useful because a number of the different proteins have benzene rings, and those benzene rings absorb UV visible light. But before we can do an analysis, we need to determine what's known as the Lambda Max, or the maximum absorbance at a particular wavelength. So here I've got 3 different amino acids with all having a benzene ring. So they all absorb light in the ultraviolet visible region of the spectrum. Because they are all different amino acids, they all absorb at slightly different wavelengths of light. So we could pick that wavelength to try and determine the concentration of that amino acid, which is called the Lambda Max. So on this graph here, you can see that our 3 amino acids, they all have a bit of an absorbance very low in the ultraviolet visible region, around 210 nanometers. But they also have some absorbances a bit further up in the UV region. So depending upon which amino acid we're analysing for, we would need to choose the right wavelength to determine that particular protein. Once we've worked out the Lambda Max, what we need to do is make up a series of standard solutions. So we go and get the proteins, we dissolve them up in water and come up with a series of standards, and then we pass them through our UV vis instrument and it produces a digital reading of absorbance, how much the sample is absorbing the ultraviolet light. Once we've got our samples and we've determined the absorbance, we plot them on what's called a calibration curve. A calibration curve is a absorbance versus concentration plot where we can read the unknown off the graph. So once we've got our unknown, we can use the graph to determine our amount. So here's an example. A 2 centimeter sample of a protein was diluted with a buffer to 25 centimeters cubed and then analysed. The absorbance of the analysed solution was 0.310. Determine the concentration of the protein in this original sample. Now in this one, they've done a dilution. So we need to account for that dilution. So we had a 2 ml sample, and then they made that up to a 25 ml sample, and that's what was analysed, centimeters cubed, sorry. So we have a dilution factor here, and our dilution factor is always the bigger one divided by the smaller one. Here, our diluted sample has an absorbance of 0.310. So what we do after establishing our calibration curve is we go to our graph and we draw a straight line from the absorbance across to the curve and then down to the concentration. So we can determine the concentration of proteins in the diluted sample by reading it off the graph. Now this was approximately between 0.4 and 0.5. Now I'm just taking a relatively middle value here just to make it easy, and I've said the diluted sample is 0.480. Now that's the concentration in our diluted sample, but our original solution would have been much more concentrated because there was a dilution. So what we need to do here is take into account our dilution factor. So what we do is we grab our diluted sample, 25, and divide it by our original sample, which was 2. So our dilution factor was 12.5 times. So our diluted sample is 12.5 times less concentrated than our original sample. So to work out the concentration in the original sample, I get my dilution factor and multiply it by my concentration, which gives me 6 millimole per decimeters cubed of protein. This type of technique is an example of Beer-Lambert's law, and Beer-Lambert's law is given in the data book. There is actually an equation for this, but essentially what we're doing is we're saying that the absorbance is proportional to the concentration. The higher the absorbance, the greater the concentration. The formula is in there. I feel that you would probably get something like this, a calibration curve, and use it. But basically his law says the greater the absorbance, the greater the concentration. Okay, some top tips for volume 7. Typical exam questions on Km and Vmax, so make sure you know the definitions of those. And then to set up a protein assay, you need a wavelength stand, you need standards, you need a calibration curve, you need the absorbance, and then you use the Thanks for watching, guys. Don't forget, drop a like on the video, subscribe if you're new, and I'll see you